Hello boys, welcome back to another interesting session of video tutorials. This session is meant for class 11th biology students and this is meant for 10th August 2020. Our memory always helps us in reflecting upon what we have been discussing and conceptualization. We were talking about the functioning of photosynthesis, how exactly photosynthesis takes place. And we said in our previous session that photosynthesis takes place through two broad phases, light phase and a dark phase. And in the light phase, which is discovered by a scientist, can you name him? The Hillman reaction. Okay, Hillman has proved beyond point that it is hydrolysis, H2O splitting up, oxygen gets released, and in light reaction many products are also formed like ATP, NADPH and O2. These are the products of light reaction. Oxygen of course comes out of the plants, diffuses out of the chloroplast, and we are all taking that oxygen. While ATP and NADPH are used to drive, so these are the energy rich compounds. I said currency of cell is ATP, used to drive the process leading to the synthesis of sugars. So ultimately the aim of the photosynthesis is to prepare starch, and starch is a polymer and the monomers are sugars, glucose. So it is this phase, biosynthetic phase, or we say dark phase, discovered by Blackman. So in this biosynthetic phase or black phase, dark phase of photosynthesis, starch is prepared. That's why it is very important phase. This process does not directly depend on the presence of light. That's why we are calling it as dark phase. But it is dependent on the products of the light reaction. It depends on ATP, it depends on NADPH, and of course CO2 must be there as a raw material, and H2O. And we must be thankful to a great scientist by name Melvin Kelvin. After World War II, worked with radioactive C14, radioactive isotopes, studied algal photosynthesis, which led to the discovery that the first CO2 fixation product, at the end of when the CO2 gets fixed, the first product is a 3-carbon organic acid. It is called 3-PGA. PGA refers to phosphoglyceric acid. So you must remember PGA, phosphoglyceric acid. And he explained complete biosynthetic pathway and named that as Kelvin cycle, because all the changes take place in the form of a cycle. And finally the glucose will be synthesized. That's why it is called Kelvin cycle. It is named after him. Further work of scientists led to the discovery of another set of plants in which first stable product of CO2 fixation is 4-carbon. There it is 3-carbon. But scientists proved that there are plants that undergo carbon fixation and the product will be, instead of 3-carbon, 4-carbon product. And there it is PGA, here it is OAA, oxaloacetic acid. Accordingly, these plants in which the first product of CO2 fixation is PGA, which is a 3-carbon one, we call it C3 pathway, or the plants are called C3 plants. And those in which the first product was C4 or oxaloacetic acid, we say C4 pathway, or we call it C4 plants. So based on the name of the product and the number of carbons present in carbon fixation, we have got two types of plants in the world, C3 plants and C4 plants. The process of photosynthesis starts with when CO2 is accepted by a primary acceptor. There should be a primary acceptor molecule. The primary acceptor molecule is a 5-carbon ketose sugar. It is called RUBP. Expansion of that, normally you will ask you, please keep remembering this, RUBP, ribulose biphosphate. I'll repeat, ribulose biphosphate. So in Kelvin cycle there are 3 stages. One is carboxylation, second is reduction, third is regeneration, CRR. Remember my name? Carboxylation, reduction, regeneration. And the entire Kelvin cycle and his co-workers proposed Kelvin cycle with these 3 steps. They showed that the pathway operated in a cyclic manner. As I said, RUBP was regenerated again and again. The Kelvin pathway occurs in all photosynthetic plants. It does not matter whether the plant is a C3 or C4. So first step is carboxylation. As the name suggests, carboxylation is fixation of CO2 into a stable organic intermediate, where CO2 is utilized for the carboxylation of RUBP, ribulose biphosphate. This reaction is catalyzed by an enzyme called RUBP carboxylase. We call it Rubisco also, which results in the formation of two molecules of PGA. So on one side you have got one 3-PGA, on the other side 3-PGA, phosphoglyceric acid. The enzyme also has an oxygenation activity, so it is also called Rubisco. What is that Rubisco? RUBP carboxylase oxygenase. Since it is an enzyme, the name ends with ASC. So Rubisco is the enzyme, and it is a must in step carboxylation. What about second step? It is reduction. So series of reactions that lead to formation of glucose involves utilization of two molecules of ATP. Two molecules of ATP will be utilized, and phosphorylation, two molecules of NADPH for reduction per CO2. So we can see the same thing in the picture also. So it is the utilization of two ATP and NADP. The fixation of 6 molecules of CO2 and 6 cycles in the terms are required for the removal of 1 molecule of glucose from the pathway. And in third step, regeneration of CO2 acceptor molecule, that is RUBP, is crucial if the cycle is to continue uninterrupted. So only RUBP keeps on getting regenerated. The regeneration steps require 1 ATP for phosphorylation to form RUBP. So ATP utilization is a must to regenerate continuously RUBP. For every CO2 molecule entering the Kelvin cycle, 3 molecules of ATP and 2 molecules of NADPH are required. This is the energy requirement. Same thing we can see here in the Kelvin cycle. Look at that. First, it is the Rubisco ribulose 1 biphosphate. So CO2 plus H2O. CO2 from the atmosphere. Step number 1, carboxylation. Because of that carboxylation, there will be 3-PGA, and of course there will be 2 molecules of 3-PGA. So 3-PGA and another 3-PGA. 2 will be there. So they keep on alternatively circling themselves. This becomes 3-phosphoglycerate or 3-phosphoglyceric acid. Both are same. And then second step is reduction. In the reduction, you can see the ATP is getting converted to ADP. That means utilization is taking place. And similarly NADPH. So this results in formation of triose phosphate. And triose phosphate will lead to formation of glucose. Glucose is a monomer. Sucrose is a monomer, and sucrose combines and forms a polymer, which is starch. And to regenerate this RUBP, regeneration step, ATP will be utilized again to form ADP plus, of course, inorganic phosphate. And this utilization of ATP ensures that Rubisco will be there continuously. As a result, in the cycle, Rubisco keeps on getting produced, and as long as Rubisco keeps on getting regenerated, plants will prepare starch. And a plant in which this Kelvin cycle takes place and the byproduct is 3-PGA, or the first product of this is 3-PGA, 3-carbon chain, then we say such plants are C3 plants. And I want you